and good morning to Professor Thomas Kuiper. Welcome to guest lecturing under 3-in-1 program of Organic and Biofertilizer Technology. It's been a great honor for me, Novalia Kusumarini, to welcome you here. Before we go to the program, I would like to sincere our gratitude to the Excellencies, Head of Soil Science Department, University of Brawijaya, Dr. Syahrul Kurniawan, lecturers of Soil Science Department, guest lecturer from Wageningen University and Research, Netherlands, Professor Thomas Kuiper, and all the audiences who follow this lecturing both from Zoom meeting and YouTube. Ladies and gentlemen, our guest lecturing will be held in 2 days under the topic of Mycorrhizae. This guest lecturing is attended by the student of University of Brawijaya, the student of University Putra Malaysia, and Thai Nguyen University of Agriculture and Forestry Vietnam, who are running a transfer credit program in University of Brawijaya. This afternoon, we will have a great opportunity to learn about mycorrhizal symbiosis, since mycorrhiza is a mutual symbiosis between fungi and plant root, which will be beneficial for plant growth and its production. And we are very lucky here that Prof. Kuiper, Professor of Soil Biology from Wageningen University and Research Netherlands, is joining with us to explain about mycorrhizal symbiosis. Before we start the program, I would like to greet Professor Kuiper. Professor Kuiper, welcome to University of Brawijaya in Malang, Indonesia. Thank you for your time, giving us a great knowledge and experience today. How do you do, sir?
I'm doing fine, thank you.
It's such a pleasure to have you here. Before we start the presentation, allow me to invite Head of Soil Science Department, Dr. Syahrul Kurniawan, to give the welcoming speech. Dr. Syahrul, time is yours.
Okay, thank you very much, Bu Nova. First of all, I would like to thanks to Professor Thomas Kuiper for having a time to give a lecture to our students about the mycorrhizae. My name is Syahrul Kurniawan, and I'm the head of Soil Science Department, Faculty of Agriculture, University of Brawijaya. On behalf of the Dean of Faculty of Agriculture, University of Brawijaya, we are say to thanks very much to Prof. Kuiper that will give a guest lecture today and also tomorrow, and with the very interesting topic related to mycorrhizae, and it will be very useful now for improving soil fertility and also, as well as biofertilizer. And also, I'm also thank you committee, Ibu Uma, and from the International Office of Faculty of Agriculture, and other organizing committee who preparing the guest lecture today. And also thank you to all students that will be that are joined in the guest lecture today. I hope the student attend and also allow the guest lecture until finish, and because the material of the course today is very important related to fertilizer and soil fertility, also for the organic or biofertilizer management. I think that's all for me. Last, Again, thank you very much for Professor Thomas Kuiper. Assalamualaikum warahmatullahi wabarakatuh, and good afternoon or good morning in the Netherlands. Yeah. Thank you, Nova.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Syahrul. Moving forward to the main agenda, we are really happy to learn about mycorrhizal symbiosis. As I mentioned before, the mycorrhiza is mutual symbiosis between fungi and plant root, which is beneficial for agriculture. Today, we will find the answers how fungi can symbiosis with plant roots and what are the benefits of its symbiosis for each organism. But first, allow me to read the curriculum vitae of our guest lecturer. Professor Thomas Kuiper is the professor of soil biology, Wageningen University and Research in Netherlands. His expertise are in soil fertility, soil biology, mycology, terra preta, mushroom, mycorrhizae, fungi, latosols, and soil ecology. He has a lot of publication, and those three are his recent publication. And also his H index on Scopus is so great. He got 46 for H index on Scopus. It is a very great inspiration for all of us. Professor Kuiper, I'm sure that everyone is waiting for your presentation. We have time until 16:14 Indonesian times or 11:14 Netherlands times for listening to your presentation and discussion. For discussion session, to all of the audience, please raise your hand by clicking raise hand icon in your Zoom room or type your question in chat room. Then I will mention your name and you can start asking your question. For the audience who follows this lecturing from YouTube, you can type your question in comment room and I will read this to get the answers from our guest lecturer. During the presentation, please stay mute your microphone. Now it's time to listening to the presentation. Professor Kuiper, time is yours.
Thank you very much, Novalia, and good afternoon, ladies and gentlemen. And let me start with sharing the screen. Is this visible?
Not yet. Not yet, sir.
Let me see what I can do otherwise. It says I'm sharing my screen, so let's hope that it will show up in a few seconds. You still cannot see it?
I can view your screen, but I can't see your presentation yet.
You cannot?
Yes, I cannot see your presentation yet.
Okay, because it says I'm screen sharing, so let me see what is wrong. Well, maybe I stop sharing and I'll try again. 1 second, please. I apologize that I still have problems with screen sharing and I don't know what's going wrong.
Or probably the committee can help you to share your screen, to share your presentation, I mean.
Yeah, that would be nice if someone could offer some support.
The committee, would you please to help Professor Kuiper to share the presentation? Thank you.
Shall I otherwise send my presentation by mail so that it can be shown through your screen? Would that be an idea?
Actually, I can see your desktop, but you still not open your presentation.
With me, it's open, so that's why I'm wondering what's happening. So you can see my desktop, but not the presentation?
Yes, I can see your desktop, but not your presentation.
That's curious. Okay, let me try once more for the last time. It now says that I'm seeing your screen, so this should work now.
It is the presentation from our committee. It is the screen sharing from our committee. Would you please to use it for your
Oh,
presentation?
it's a PDF. Then I will just use a PDF. That should work well enough.
Yes, thank you very much.
And then I can go down. Now I have to ask next slide by you, but that doesn't matter. Okay, well, good afternoon again, ladies and gentlemen. And I apologize for having some problems with the Zoom, but I'm glad for the invitation to lecture for such an international group of students, which I understood are from 3 universities. And my plan is to lecture about mycorrhizal symbiosis today, and then more specifically deal with issues for the application of mycorrhizae in agriculture tomorrow. Can I have the next slide, please? So let us start here in this beautiful place, which you all know as Kebun Raya Bogor, or as it was called at that time, the Botanical Garden of Buitenzorg. And the reason for starting with this picture can be seen in the next slide, where you can see on the left an old-fashioned scientist called Professor Janse, who was a professor of biology who worked at that time in Bogor. And he was interested in plants, and he also was interested in roots. And so when he looked under the microscope of the root, in this case of a species of Pandanus, he saw all these fungi that you see on the upper right. And what he was drawing at that time, and that was before the invention of all the beautiful stains we currently have, is quite resembling what you could see on the lower picture below. Next slide, please. And so Jansen wrote in French, but I have translated it for you in English, that several scientists had noted the presence of a fungus in the cells of the cortex, so the outermost parts of the plant root, and that they have seen, and that's what he saw as well, that not only are those fungi alive, but that the plants are not showing any signs of sickness. And then he asked the question, what could the presence of the fungus indicate? Next slide, please. So these were a few of the questions that Jansen asked in the paper he published more than a century ago, actually from 1907. And so the first question is, are these fungi parasitic? And Jansen said, no, this cannot be the case because you see those fungi on plants that seem healthy. And the next question that Jansen asked was, is there something peculiar to his plant species, the tree Pandanus, or is it more common? And then he knew from his observation that they occur in almost all plants that they studied. And that raised, of course, the main question. If these fungi are not parasitic, not harming the plants, what are they doing? Are they just being there, hanging on, or are they having some benefits for the plant? And Jansen knew the literature that these organisms, which he called root endophytes, which just only means organisms inside plant or inside the root, that there was literature from Europe, especially by a German scientist called Frank, but also a French scientist called Gallo, and that for them they had the term mycorrhiza. And next slide, please. That is the topic of my lecture, mycorrhiza, which is a relationship between roots and fungi. And that relationship is of benefit for both parties. And that relation is based on an exchange, or if you want, a trade of resources that are limiting one organism over the other, over what's called a symbiotic interface. So there is a kind of common structure inside or between the cells over which that trade takes place. And then a major question is, what do each partner gets, and we know that the plant gets nutrients and that the fungus gets carbohydrates or energy. And the main question that has not been resolved is the extent to which that exchange is under control of the fungus or of the plant. People have been proposing mechanisms by which plants can prevent fungi from what we call cheating. Other people have provided evidence that the exchange and the exchange rate is more under the control of the fungus. I will not dwell with it because that is still an issue of debate. But let's go to the next slide. And here you can see an example that this relationship is beneficial. If you compare row number 1 at the left with row number 3, then you see the same plant. This is Japanese bunch onion without on the left or with Mycorrhizae on row 3. And you can start fertilizing your plant. That's what you see in row 2. Without and with Mycorrhizae that you see in row 4. And again, you see that the plants benefit from this symbiosis. So the benefit for the plant is quite evident here. Next slide, please. And also evident here. On the left, you see a soil that has been sterilized, fumigated, so where most soil organisms have been killed. And on the right, the field has not been treated, and so the mycorrhizal fungi are there. And again, you can see much larger plants. So again, showing that the relationship is of benefit to plant. Next slide, please. So the question then is, how does mycorrhizae work? And for that, we need to go to soil science to understand the behavior of certain nutrients. We know that in many soils, and that is even more the case in tropical soils, the availability of some nutrients is very low. And that is especially the case for phosphorus. And it's not because there is very little phosphorus in the soil, but because most of the phosphorus is very strongly bound to mineral soil particles. The tropical soils are red, they contain iron. Iron is positively charged, phosphate is negatively charged, and so there is a strong binding, and there is only very little in the soil solution. Plants are very efficient in taking up that phosphorus when it comes close enough to the root. So there is a very slow movement of phosphorus towards the root and a very rapid uptake as soon as it's close enough to that root. And that results in what we call here the phosphorus depletion zone, a zone where the concentration of phosphorus is very low because everything within that zone is immediately taken up. But that zone is surrounding by an ocean where there is still some available phosphorus in the soil solution. But that moves only at rates of millimeters per hour or even less, it's not easily accessible. Well, you can measure that depletion zone. The next slide, please. And here you can see what has been modeled, but you can also measure it. And you can see that just at the root surface, so at the left of the graph, where the distance is 0, concentration of phosphorus is very low. And only 1 millimeter of the root, there is still enough phosphorus around. And so plants are faced with the question, how can it overcome that problem of the depletion zone? And of course, you can make more roots, but you can imagine if this distance is only 1 millimeter and the root is 1 millimeter thick, you need to make lots and lots and lots of roots. So that's not how plants have resolved this. Next slide, please. So one way by which plants resolve this is by making root hairs, as you see on the picture. And if you have a germinating seedling on a petri dish, you see all those most straight root hairs. If you would collect the root hairs in the soil, you see that they are bent and very irregular because they are flexible and so have to adapt their way through grains of sand and small stones and things like that. But they are very important for nutrient uptake. Next slide, please. And here you can see how it works. So on the left, we saw what we just saw before is the depletion zone and a lot of available phosphorus. On the right, you can see that as soon as a plant has mycorrhiza, this is an old slide where mycorrhiza was still called VAM, where you can see at least that thanks to the root hairs, this depletion zone, the zone over which the phosphorus can be acquired, has been tremendously increased. Next slide, please. And so you can just model how root hairs would work. And what people have found out is that if you do not include root hairs in your model, as we can see on the left upper graph, there is a large mismatch between what the models show in terms of nutrient uptake and what the actual uptake is. The dashed line, the upper one, is the one-to-one line, which predicts uptake as same as observed. But you can see for several plants, and especially the bold line with the filled triangles below, called Russian thistle, that the observed uptake is 3 or 4 times higher than what you predict. And as soon as you include root hairs, which you can see on the graph on the right below, and then again you have the one-to-one line between predicted and observed uptake, and then you can see that there is a very close match. So having root hairs is one way by which plants solve the problem. And so root hairs extend that depletion zone, and we can see that in the next slide. Where we can see different varieties of barley, a major grain cereal, and where you can see again that the concentration of the bulk soil solution of 0.45 millimoles phosphorus is reached now not at 1 millimeter, but at around 2 or 2.5 millimeters around. And you can see, if you compare the two varieties, that the one called Zeta has earlier reached that plateau than the variety called Zalka. And that means that Zalka has slightly longer root hairs. Next slide, please. So that is one solution that plants have, make root hairs. But there are still limits to root hairs. You can see they can still only go 1 millimeter additionally outside of that depletion zone, because that is the length of root hairs. Exceptionally, they are 1 to 1.5 millimeters. Sometimes they are even shorter than that, and so there is a limit to that. And so plants have figured out another solution to that problems, and that is by not doing this yourself, but by association with those beneficial fungi, and that is the mycorrhizal symbiosis. Next one, please. So this is then the solutions plants have. Solve it within the plant, the in-house solution, or do it yourself, or what's called outsourcing and leave it to others. And that you can call a little help from my friends, as this famous song by the Beatles is about as well. And we know, and I will come back to that later, that this symbiosis is more than 400 million years old, and still plants rely on that symbiosis. Next one, please. And so here you can see what happens if you have not just, again, this non-mycorrhizal root, but here you have the mycorrhizal fungus. You can see it can move way outside of that root and again enlarge that depletion zone and take up more phosphorus. And if you think about this picture for a second, you can imagine that if a fungus makes more of those external structures called hyphae, by which they grow outside of the root, if you make more of them, you would be able to acquire more phosphorus. And that's what the next slides will show. Here you see on the horizontal axis the amount of hyphae that are being produced. And those numbers are the length of the mycelium in meters per gram of soil. So if you have a gram of soil, which is very little, that's just a piece of 1 centimeter by 1 centimeter by 1 centimeter, so very little, then you can easily find 10 or 15 or 20 meters of those very thin fungal hyphae. And you can see for two different fungi in this case, that if there is a larger length of the hyphae, so a larger size of the whole network that the fungus provides, there is more uptake of that phosphorus. So plants, as I said, have two solutions. Either do it yourself by making long root hairs, or make mycorrhizal associations, or make combinations of those, as the next slides will show. So on the left, you see a plant without mycorrhiza. On the right, you see a plant with mycorrhiza. And you can see that even in this specific case, where the plant root is quite branched, which also helps, has quite long root hairs, there is still some additional benefits of mycorrhiza. Next slide, please. And so people have been investigating how root hairs and mycorrhizas work together. And if you want to do that, you need plants of which you have variants or mutants that have long or short root hairs. Well, on the left, you see the 3 mutants with long root hairs. On the right, you see 3 mutants with short root hairs. The black ones are the ones with mycorrhiza. The white ones are the ones without mycorrhiza. And so you see in all 6 cases that there is more phosphorus in the plant with mycorrhiza plus AM than without mycorrhiza. You also see that if plants have very short root hairs, there is a very large effect of mycorrhiza. If the plants have longer root hairs, there is a smaller effect of mycorrhiza, but still there is a positive effect, as the black bars are always higher than the white bars. So yes, making long root hairs helps, but making mycorrhiza as well does help additionally. Next slide, please. So here you can see again this importance of root hairs. On the left hand, you see a clover plant, which has fairly short root hairs of only 0.2 millimeters. And you can see that in that case, there is a large benefit of mycorrhiza, which is in the orange bars compared to non-mycorrhiza. On the right, you have a grass, which has root hairs of about 1 millimeter. And here, the effect of mycorrhiza is much small. So that raises a question that people have long been discussing and for which we are not 100% sure we know the answer. Why do plants not all make long root hairs? Why are there still plants with having root hairs of only 0.2 millimeters if other plants have root hairs that are 5 or 6 times that long? Next slide, please. And here we see what is part of that explanation. We know that plants that have thick roots, and that's what you can find in certain tropical trees, where the roots can be 1 or 2 millimeters thick, and that they usually have few root hairs and that are very short. If you would just plant an onion in your soil, then you would see that onion has few roots, has thick roots with very short and only sparse root hairs. And if you would plant a cereal like wheat or barley, you would see thinner roots with larger and longer and more abundant root hairs. And we know that the plants that have the thicker roots, as you can see from both slides, and note the right slide has the thickest roots on the left and the thinner roots on the right. The plants that have thicker roots have more mycorrhiza. So the balance between root hairs and mycorrhiza depends on the thickness of the root, and because that's correlated with the abundance and the length of the root hairs with those root hair properties as well. Next slide, please. So we know that, as I said before, plants with more root hairs have also longer root hairs, that plants with thick roots have few and short root hairs. And the question is, why is that the case? And one suggestion that has recently been offered is that root hairs are very short-lived, roots are much longer-lived, and you can also see that the duration of roots, so the root longevity, is larger with thick roots. The above picture plots the lifespan of roots on the x-axis and the root diameter on the other. And you can see that thick roots of about 1 millimeter or more can easily live 1 or 2 years. And the thin roots on the left of the upper graph of 0.2 millimeter or even less usually live only a few months. And you can imagine if your root is very long-lived, it doesn't make sense to make root hairs that are very short-lived. And apparently plants are programmed to make short-lived root hairs. And so making long root hairs that pass away very soon will not very much help. So that's why we think that plants with thick roots rely on mycorrhiza rather than on root hairs, and plants with thin roots rely much more on root hairs, the length and the number, but still rely a bit on mycorrhiza, as the next slide will show as well. So here again, we see the system where on the left we have a poorly branched root. And then we see in the dashed line that it has a high benefits in terms of nutrient uptake. And the more branched the root gets, so we've moved the x-axis to the right, the less the benefit is in terms of nutrient uptake or phosphorus uptake, as we just saw. But, and I will come back to that question tomorrow when it comes about application of mycorrhiza, having mycorrhiza has more benefits than only increased nutrient uptake. And that is what people refer to as the multifunctionality of mycorrhiza. The idea that mycorrhiza does other things. And one other of those things, and we will discuss that tomorrow, is that plants that are mycorrhizal are in fact less attacked by plant pathogens, are less grazed by root herbivores. So what those plants with highly branched roots lack in terms of benefit for nutrients, that is what they get by another benefit of mycorrhiza, namely that they can protect plants against their enemies. Next slide, please. So this is then how it works. To summarize what I said before, by having mycorrhiza, plants can enlarge the soil volume over which they can take up immobile nutrients that are very well buffered in the mineral soil. And they do that by the extension of this depletion zone. This zone where the concentration of phosphorus is very low, which is the driving force of diffusion of phosphorus towards the root. You can also imagine that those hyphae, those fungal structures, are much smaller than root hairs and thereby can grow into, have access to soil pores with a smaller diameter or a smaller pore neck, and therefore can find water when the larger pores are dried out. And when you find water, there may be nutrients dissolved in that water, so again you have more uptake. We also know that the uptake system itself, so if you look at the physiology of the uptake system of the plant, that the one by the mycorrhizal fungus is more efficient than that of the plant. There are not many data, but there are some studies that show that. We have recently worked on modeling such an uptake system differences, and it explains an additional benefits of mycorrhiza. And then in some forms of the mycorrhizal symbiosis, but not in all of these forms, they can also have access to forms of nutrients in organic forms, like amino acids or small parts of proteins or organic phosphorus that non-mycorrhizal plants would not. But as mentioned, and we'll come back to that, that's only in some form of mycorrhizal symbiosis, not in all of them. Next slide, please. So these are the other benefits we will discuss. The access to smaller soil pores. We know that the plant that is mycorrhizal has a different hormonal status, so the expression of certain growth hormones like auxin or EAA or other plant hormones are changed when the plant is mycorrhizal. In some cases, they enhance the weathering of rock, which increases the availability of cations. We know they can protect against heavy metals, and in very acidic cases, they can protect against the toxicity of aluminum. And that is certainly important in those acid tropical soils where there are large amounts of free aluminum available. And as I mentioned, they can protect plants against their enemies, the pathogens and the herbivores. And we know that having mycorrhiza can help to improve the soil structure by contributing to the formation of aggregates. So this is a major slide where you can see all the benefits of mycorrhiza next to the one in the previous slide, which is about the enhanced uptake of nutrients of low availability. And then phosphorus in the tropics is the main example of such a nutrient. Next slide, please. So again, we see on the right, big plants and small plants. And again, the small plants have the NM, so not mycorrhizal. Big plants have AM, so they are mycorrhizal. And we now know from what I said before that this has to do with the enhanced uptake of phosphorus. If that is the case, you would say, why not fertilize more with phosphorus? Because then the availability of phosphorus increases and maybe plants can do it by themselves. And yes, to a certain extent, that is true. And you can see in the left upper graph that if you increase the amount of phosphorus in the soil, going from 0 to 480 millimoles of phosphorus per liter, you see a strong response of the non-mycorrhizal plants, the open circles, but you see that apart from the highest level, even at moderately high phosphorus levels, the mycorrhizal plant is much larger. And you can also see, and that is important for what I will discuss in a second, that the concentration of phosphorus, so the amount per gram of plant, is also larger with the mycorrhizal symbiosis. And that remains true even at the highest doses of phosphorus. So yes, to a certain extent, you can replace the benefits of mycorrhiza by adding phosphorus fertilizer. But phosphorus fertilizer is expensive. We know we are running out of phosphorus fertilizer in probably 1 or 2 centuries. And we know that especially in those acid tropical soils, most of the phosphorus does not end up in the plant, but gets absorbed by those mineral particles. So adding phosphorus, as you can see in this experiment over here, is something that almost all farmers, unless they are extremely rich, cannot afford. And so if you have financial limitations to buying phosphorus fertilizer, you depend on mycorrhiza. Next slide, please. So yes, and that is how we often present mycorrhiza, where you show people how much yield gains, how much more yield you can produce if your plants are mycorrhizal or non-mycorrhizal. And it tells you that at least with this plant, and this is onion, and I said before that onion has very thick roots, not that many roots, very few and thin root hairs, that almost always there is a benefit of mycorrhiza. But you could also read the upper left figure in a different way, and that's shown in the upper right corner. We all know that in order to make agriculture more sustainable, we should think about reducing the amount of fertilizers that we apply. So rather than saying how much more yield we can produce at a given fertilizer level, we could also ask ourselves how much fertilizer we could save at a certain yield. And that's what the right upper graph shows. And it suggests that you need to add something like 200 or 250 millimoles of phosphorus per liter without mycorrhiza, and you need to add hardly any phosphorus to get the same yield. And I will show tomorrow a calculation we did with maize, a very common crop all around the globe, where we tried to calculate the value of these fertilizer savings. And then again, on the 2 lower figures, you see again that not only is there more biomass, but also the internal phosphorus concentration per unit of mass is also increased. Next slide, please. And we know that especially this increase happens in plants that respond very strongly to mycorrhiza. And that is important, and we can see this in the next slide. Because when we produce food, it is both about producing enough food, so the quantity of food, but also about the quality of food. So you can look at the biomass, and I think this was with maize. You can see without and with mycorrhiza. And if you add low phosphorus doses, you can see that the mycorrhizal plant weighs between 1.7 and 2.4 grams, and without mycorrhiza, around 1 gram. So yes, you increase plant benefit, but at the highest phosphorus dose, the plant without mycorrhiza weighs 2.8, plant with mycorrhiza weighs 2.7 gram, so that's roughly the same. But if you look at zinc at the right column, and zinc is a very important nutrient for especially growing children and pregnant women, we can see that in all cases, the plants with mycorrhiza contain more zinc. So it's not only that with mycorrhiza and good management of mycorrhiza, we increase the yield, and so we have more food, but we also have a higher concentration of zinc. We also have higher concentrations of phosphorus, and so we have also improved food quality. So this was the first part about the general effects of mycorrhiza, but I have spoken about mycorrhiza as though this is one kind of thing that happens. And so what I will do now is to look a bit about the evolution of mycorrhiza and look a bit about the diversity of mycorrhiza. Next slide, please. And so this is what plant scientists and evolutionary biologists now considered as established, that we know that ever since plants became terrestrial, so when the algae that lived in an aquatic environment colonized the land, there has been this mycorrhizal symbiosis. And if we try to be even more accurate, we know that the mycorrhizal symbiosis is older than the plant roots as we know them now. Then, of course, we need to define what a plant root is, and that is a specialized organ that has what's called a gravitropic response, which means that they grow downwards rather than upwards, just like shoots. The roots have this protective root cap, have root hairs, all the things like that, and they evolve much later than this mycorrhizal symbiosis. So that is a conclusion that I drew in a recent paper and that I shared with you, and so you can read this again, that plants, when they evolved, had to take into account the constraints that were imposed by those mycorrhizal fungi. And so plants had to adapt to the fact that there were fungi already around. Next slide, please. And here you see some of those primitive plants that have more shoots or bulb-like structures that lie in the soil than have true roots. And from these, the mycorrhizal symbiosis grew. Next slide, please. So here we have such a plant of more than 400 million years old. And people found those fossils in a geological formation in Scotland called the Riney Chert. And you can see that, although it's not always very evident, so you need to have some experience, you can see already some kind of mycorrhizal structures in those roots. And we can see them better in the next slide, where we see a fossil spore of a mycorrhizal fungus. And on the lower part, we see a fossil arbuscule of around 400 million years old. And if you compare that picture of the fossil arbuscule with arbuscules that I will show later, you will see that they have not much evolved. And also the structure of the spores have not much evolved as well. And so that's what we can confidently say. If there were no mycorrhiza, then terrestrial growth of plants would have been impossible, which means that humans, because we are terrestrial organisms as well, would probably not have evolved. And that has nothing to do with water. And you would say, well, if you move from water to the land, you may have a shortage of water. But that is not a problem because many soil pores are still filled with water. But it's especially because in terrestrial environments, where you have acid, iron-rich soils, phosphorus is the main problem. Next slide, please. So if this symbiosis is around for 400 million years, then you can imagine that plants must communicate with the fungus in a very subtle way. And that's what we call the mycorrhizal dialogue, the way in which plants and fungi leak compounds in the environment that are recognized by the others. So it's organisms speaking different languages that we can call plantish or fungi, and by which the signals by one organism are taken up by the others. The plant leaks compounds called strigolactones that are recognized by the mycorrhizal fungus, and they also has what's called cutin monomers, so what otherwise make the corky layers surrounding cells that are also recognized. And the fungus makes certain nitrogen-rich sugar-like molecules that are called keto-oligosaccharides, or sometimes there are even fatty acids and next to it lipo-keto-oligosaccharides. And you can easily forget about the words, but the idea is plants leak compounds that the fungus recognizes, the fungus leaks compounds that the plant recognizes. By that way, they start communicating, so to speak, talking to each other, and that results in the mycorrhizal symbiosis. Next slide, please. And here you can see how it goes, because a plant has a problem. If a mycorrhizal fungus knocks at the door, you cannot open the door. It's the same with you at house. If someone knocks at the door, you don't know for sure whether it's a friend or whether it's a thief. So if you keep the door always open, you can be pretty sure that not only the mycorrhizal fungi will enter, but also that parasites and pathogens and other organisms will enter as well. So that dialogue that I just showed in the previous slides must do two things. It must make sure that the mycorrhizal fungus, when it knocks at the door, gets in, but it also must make sure that if the enemy knocks at the door will remain closed. And molecular biologists have spent a long time in understanding that dialogue. You can see that on the cartoon on the left and also the right upper version. So here we see on the left a plant cell with the plant nucleus just at the bottom of the cell, and at the top of the cell we see a mycorrhizal fungus getting close to that root cell. Then the fungus forms what's called an aprosorium, which is a structure by which it could penetrate the root in the end, and by that it knocks at the door. And then it starts releasing signal molecules, which are called the Myc factor. They're called DMI2 and DMI3, but you can forget about those terms. But as a consequence of this exchange, the plant cell that was first positioned at the bottom of the cell now moves towards this aprosorium. So they establish contact. It's like when someone knocks at the door and you decide to go to that door. If the signals, the way the organism knocks at your door is indicating that someone is reliable. Then with some further exchange of signaling again, the fungus can be through that aprosorium. A new structure is formed to separate the fungal material from the plant material, and you can see this in the lowest part where there is a membrane separating the fungal hyphae from the plant hyphae. That is being made by the plant, and then the nucleus moves back to its original position, which you can see in the third and the fourth figure on the left. And by making that thread and this assembly of that structure, the fungus can grow into the cell. And we called it the red carpet. You knock at the door, the plant moves its nucleus towards the door. If the signal is honest, the door is being opened, the red carpet is rolled out, and by that the fungus can enter the cell. And that, of course, is a very fine dialogue. It works at very low concentrations, and except for one very specific case, no pathogen has been able to hijack that communication. So it's still a very sure way in which mycorrhizal fungi and plants can communicate, ensuring that the fungus can enter the cell and ensuring that the enemies cannot enter the cell. Next slide, please. So that is about the kinds of mycorrhizal associations. Up to now, I have mentioned Mycorrhizae as though there is one type. We now recognize 4 major types. They're called ectomycorrhizae, arbuscular mycorrhizae, ericoid mycorrhizae, and orchid mycorrhizae. The latter 2, as the name implies, are very specialized. Ericoid mycorrhizae only occurs with ericoid plants, so plants of the Ericaceae, and orchid mycorrhizae only occurs with orchids, so plants of the family Orchidaceae. The 2 other types of mycorrhizae, ectomycorrhizae and arbuscular mycorrhizae, occur with many more plant species. And you can see, just as an indication, how many plants form arbuscular mycorrhizae and how many fungal species are able to do that. And here I've listed the number of arbuscular mycorrhizal plants at 200,000, based on the idea that there are maybe 250,000 higher plant species. Some other people think that that number is higher. Then, of course, the number of mycorrhizal plant species is much higher as well. And I would like to spend the final few minutes where I am talking, and before you have a chance to ask your questions, to show some pictures of how those mycorrhizae look like. Next one, please. Here you see the ectomycorrhizae, and you see a picture of Albert Frank, the German who was the first to use the term ectomycorrhizae. And what Frank noted was that with certain trees, you cannot see any root hairs at all, because the root is completely surrounded by what you can call a glove of fungal tissues. If you look at the drawing by Frank on the right lower part, you see the whole outer part of the root covered by this grayish material, which is all fungal material. And you can see that there is no direct contact even between the root cells and the soil environment. That any exchange the root has with soil environment must go through the fungus. Next slide, please. And so this is how ectomycorrhizae look like. This is the mycorrhizae that you can find with pines. And there are some pines in Indonesia as well. And you can see that the root is completely covered by a white sock or a white glove. That is the fungal tissue. In the case of pines, those roots are also forked, but that does not happen in every plant. In this case, the ectomycorrhizae is white, but there is a large variation in color and in form, as the next slide will show. Next slide, please. So here you can see some variation in form. They can be white, as we saw in the previous graph. They can be black, they can be brown, they can be yellow in some cases. Sometimes they are a bit violaceous. Sometimes they are smooth, as the right upper part. Sometimes they are very hairy, as the left upper part. Sometimes they are in very large groups close together. And sometimes they have those thick strands or thick cords or rhizomorphs for which they can really achieve long distance transport. Next slide, please. So here you see an electron microscope picture of a very young root, and you can see that the root is completely covered by those fungal hyphae, as we saw in the drawing before. And so where you can see if this fungus is completely covering the root, there is no direct interaction between the plant cell and the soil environment, and that any exchange must go through the fungus. Next slide, please. So here you see the two characteristic figures. On the left, you see the sock or glove, or whatever you would like to call it. Technically, we call it the ectomycorrhizal sheath or ectomycorrhizal mantle, so the outside structure. And then you have fungal hyphae between the root cells. They're always between, never in root cells, and that's called the Hartig-net, named after another German student of Mycorrhizae more than 1 century ago. Next slide, please. I said that those hyphae in the case of ectomycorrhizae are between and never in the cells. You can clearly see this here, where these cells are a bit pushed away, creating space for the fungus, but there is no fungal material in the cell. And we will see in a second the other type of mycorrhiza called the arbuscular mycorrhiza, where it is present in the cells, as we saw before, how the plant arranges that. And that's why you see in older publications still the term endomycorrhiza. But because endomycorrhiza combines this arbuscular mycorrhiza and the ericoid mycorrhiza and the orchid mycorrhiza, and because these are very different kinds of mycorrhiza, they are usually not anymore taken together under the term endomycorrhiza. But if you have all the textbooks, you may see that word, but often it indicates that the fungus is in the root cell, but it's good to ask yourself which mycorrhizal type it is. Next slide, please. Well, as a plant scientist, you may be, of course, most interested in what's happening on the roots, where you have this fungal sheath or this fungal glove, where you have the fungi inside the roots. But as a soil scientist, and if you're interested in the uptake of nutrients, the main interest should be on the hyphae that go outside from the roots into the soil. And here you see two ectomycorrhizal fungi, and you can see, and of course it's a bit due to the specific experimental condition, but you can see that there is a large amount of fungus, that it really goes very far into the soil, and that's all there to support this small plant. Next slide, please. So then we go to arbuscular mycorrhiza, and one major difference is that ectomycorrhiza can be seen with the naked eye. These root tips surrounded by this fungal mantle. You can easily see that if you dig out a pine tree. If a plant has arbuscular mycorrhiza, you cannot see this just by digging out plant roots, although often the roots are a bit more yellow. But it's not always easy to see. So if you really want to study arbuscular mycorrhiza, you need a microscope. We saw some of the microscope views on the MP4 presentation that was shown before. Next slide, please. So here we see this arbuscular mycorrhiza. There we see a number of the internal structures called vesicles, called arbuscles, called intracellular hyphae. And then we have the entry point where there is a specific structure, where the fungus knocks on the door, called the aprosorium. And all these structures are only found in the cortex, so the outermost root cells. The central part, called the stele, is always free of mycorrhiza. Next slide, please. Here we see the arbuscles. These are hyphae that branch and branch and branch ever more finely. And this is the structure where the exchange of phosphorus delivered by the fungus against carbon delivered by the plant takes place. And of course, if you have all the expensive equipment and you can make beautiful pictures, arbuscles look like this. But the way we normally do it, when we cook roots, when we stain them with the blue stain, they look like on the next slide. Next slide, please. And where you see this somewhat fake, previous one, please. Here you see the vesicles, they stain blue. The previous slide showed the arbuscles. And they are not blue by themselves, they're only blue because we stain them with that compound. Next slide, please. So here again, you see this arbuscle and the vesicle stained blue, and that's what you can see if you look inside the root. And that's, of course, what we need if we need to determine how much mycorrhiza is there in the root. But of course, in order to do the job, you need the fungal mycelium, the fungal network outside the root. And you can see that on the lower picture, where all the very thin threads are all fungal hyphae, and you can see how much thinner fungal hyphae are compared to plant roots. Plant root may be 100 or 200 micrometers diameter. The fungal hyphae may be 2 or 4 or 5 micrometer in diameter. So yes, you can make far more material in terms of hyphal length than you can make in terms of root length. And you can also enter small pores much more easily. Next slide, please. Only to show, this is the kind of symbiosis you see with orchids, where the root cells of the orchids are completely filled with fungi. And the orchid mycorrhiza is a bit special, because if you have ever looked at orchids, you will see that orchids have extremely small seeds. And so the fungus does not only provide nutrients, but in the seedling stage, the fungus also provides carbon. And in fact, you can say that with orchid mycorrhiza, in the seedling stage, the plant, the orchid, is parasitizing on the fungus. Only when the orchid is adult and has green leaves is there some benefit for the fungus, although it's not always that big. Next slide, please. So then we go to this ericoid mycorrhiza, which, as I mentioned before, can only be found in members of this one plant family, which means in the tropics there are plants of the genera Rhododendron and Mycorrhizae, where you can find them. Again, the cells are completely filled with fungal material. But as rhododendrons do not play any role in agriculture, I will not further talk about it. Same is true about orchid mycorrhiza. Of course, if you're an orchid grower, you must know how to manage them. But if we want to focus on applications of mycorrhizae in forestry and agriculture, it is enough to look at the ectomycorrhizae and the arbuscular mycorrhizae. Next slide, please. So that's the question then, where we can find them. We find ectomycorrhizal plants in plantations of pines and eucalyptus, and we can find them in the native forest of Dipterocarpaceae, the Meranti trees. In almost any other habitats, grasslands, cropland, and in other tropical rainforests that are not dominated by Meranti, you find arbuscular mycorrhizal plants. And then you find non-mycorrhizal plants in some extreme situations and in cases where the soil is very much disturbed. And of course, when we humans apply agriculture, and we will come back to that tomorrow, then we disturb the soil a lot by plowing, and that may have consequences for the way we manage arbuscular mycorrhizae. But as said, we will deal with that tomorrow. Next slide, please. So that's what the plant benefits. And so this is what the fungus benefits. This fruit body of this mycorrhizal fungus is completely formed of carbon that is provided by the tree. And you can imagine that people wondered about the amount of carbon that the plant has to deliver, and the question whether that is or is not expensive, and whether a plant really benefits from mycorrhizae. And that's, of course, an important question in agriculture. Next slide, please. So here you see a case where the plant definitely does a better job without mycorrhizae, called NM, than with mycorrhizae, called AM, with two different plant species. And we know that there are several of those cases. And I will not, because of the time, dwell with the mycorrhizae, so we can skip the next slide and we go to the slide after that. So you can move 2 slides. Next one. So here we have the question, how come that some plants respond negatively? What you see in many textbooks is that it has to do with the amount of carbon that comes from the plant and goes to the fungus. But we have shown that that cannot explain that large growth response. So there are different other factors which may have to do with the fact that the fungus itself, of course, also needs nutrients and may not give everything to the plant. It may change some other plant responses. It may change the architecture of the roots. And so there are many cases where plants are not responding to mycorrhizae, although I would say this is the exception rather than the rule. What is normal in agriculture is like what you see above with clover, not what you see below. Next slide, please. So what I said in the slide before, in most cases you see that plants benefit. Almost all plant species form mycorrhizae in one form or another form. And that is not only the case for the wild plants we can find in the forest, but also for almost all of our agricultural crops. So if you want to work on sustainable agriculture, you should never forget that our agricultural crops are forming mycorrhizae and that the plants could not grow well without mycorrhizae under conditions of normal soil fertility. Of course, we have increased the amount of fertilizer, we have enhanced the soil fertility, then plants became less dependent on them. But of course, if we want to make agriculture more sustainable, we cannot continue doing so. And it's not only that the plants cannot complete their life cycle and form healthy seeds without mycorrhiza. We also know that the mycorrhizal fungus cannot complete the life cycle without plants. We cannot grow mycorrhizal fungi on a petri dish like you could do with other fungi that you grow under such conditions. Next one, please. So then the question is about plants that are not forming arbuscular mycorrhiza, and then plants can form different roots. There are all kinds of beautiful root systems, but you don't find them in crops. You can, of course, become a parasitic plant if you then grow on trees or whatever else. You don't need roots to take up nutrients because you suck them from the tree. You can become a carnivorous plants like those pitcher plants, Nepenthes, then you can also do without it. But we know that even the plants that do not form arbuscular mycorrhiza, they still can, under some conditions, grow with mycorrhiza. They still have maintained this dialogue system with the fungus. But yes, they can also grow in habitats where we have, due to human activity, to disturbance, etc., about which we will speak tomorrow, grown without it. But they would often, especially in close vegetation, have a problem when growing without mycorrhiza. And that is because you do not only have to assess whether a plant grows well with or without mycorrhiza, and we saw many of those plots with one plant species with or without it, but in most vegetation, you have also to take into account what your neighbor does. And if your neighbor is mycorrhizal, it's very hard for a non-mycorrhizal plant to survive. And that brings me to my final slide and the take-home message for today. And I think that is something that we tell our students as soon as they start studying in Wageningen ecology, that plants, and that's true also for the crops and agriculture, they do not have roots as just something, an organ that is only made by plants, but they have mycorrhizas, so roots together with the fungus, and it's that combination that is crucial for plant function. So if you have kept from this lecture this final take-home message, that if you grow plants without mycorrhiza, you're already looking at an artificial system, because plants in nature always have mycorrhizas rather than only plant roots without fungal stuff. Then that's a major message that I want for today, because understanding that message is important for what I would like to discuss tomorrow when it goes about the management of mycorrhizas in agriculture. And I thank you for your attention, and I apologize again for some of the problems I had with the Zoom, and hopefully we can fix it for tomorrow. And I'd be happy to take any questions.
Yes, it is very well presentation. Thank you very much, sir. From your presentation, I can highlight that mycorrhizas is not parasitic to plant and having some benefits for the plant as well as for soil structure. Plant receives nutrient from mycorrhiza, and fungi receives carbohydrates from plant roots. And also plant growth, production, and quality will be better if it is associated with mycorrhizas than other which is not. And the last is plant in nature always has mycorrhiza. It is quite interesting to have discussion about these topics. And now we are in discussion session. But before starting the discussion, I please to all of the audience to take a quick break. Probably you need some water or etc. And anyone who want to ask some question, please raise your hand or type your question in comment room. And for the committee, please collect the question from comment room in YouTube, because this guest lecturing are live streaming also in our YouTube channel. Oh, okay. We have one question from Anita Destranti. The question is, how do plant hormones such as auxin, gibberellin, cytokinin modify mycorrhizal functioning? How does the mechanism? Is it beneficial for plants or the opposite? Does the mycorrhizal compete with plant in plant hormones acquisition? Professor Kuiper, please answer the question.
Well, I'm not a molecular biologist, so exactly how it does modify that mycorrhizal functioning is not always known. What we know from one specific hormone, which is called this class of strigolactones, is that plants leak them in the environment, and by that the mycorrhizal fungus is attracted, and when the plant is colonized, it goes down. In other cases, we know, if we compare root systems, that plants with mycorrhizae usually have more branched roots than plants without mycorrhizae, which means that the plant hormones that are changed make plant roots branch more and grow less in terms of what we call the exploration mode, getting further away. And of course, having more branch roots is of benefit for the fungus, because then the roots stay close by and the fungus don't need to grow as rapidly as the root does. It may be beneficial for the plant as well, but in some cases, the plant can explore less territory. So usually it's of benefit for the plant as well, those hormonal modifications, not always. And when it comes to drought resistance, so tolerating periods of drought, we know that mycorrhizal fungi alter photosynthesis, alter what's called stomatal conductance, and that means that plants can still photosynthesize a bit when it's dry, when they're mycorrhizal, while they would stop photosynthesis when they're not mycorrhizal. So again, that is of benefit for the plant. In the case of arbuscular mycorrhiza, we think that it's just the fungus who sends signal that modifies the plant hormone status. With ectomycorrhiza, we know that the fungus themselves can produce the same hormones, the same auxins as plants can, and that they thereby modify the plant system independently.
Thank you very much, Professor Kuiper. Another question, probably the audience who follow this guest lecturing from Zoom meeting. Okay. Another question from Dayat Purnomo. Do mycorrhizal have a host-specific preference? An example of a beneficial role of mycorrhiza is with suppressor. How can mycorrhiza specifically suppress without suppressing crop growth and developments?
Well, I would first compliment the person who asked the question, because it's a very good question, because we always talk about the benefits of mycorrhiza, but the example already suggests that sometimes the world is a bit more complicated. So let me try to summarize what we know, that most mycorrhizal fungi, if they are forced to associate with the plant, so if you put a fungus together with the plant in a pot, they will make a successful symbiosis. If you allow plants and fungi to choose, then they are a bit more selective. So if you would sample mycorrhizal fungi in a field with mixed crops, you will find to some extent different fungi with different plants. There has been a whole discussion on, or at least some people have studied, the role of mycorrhiza in weed suppression. And then, of course, the issue is how come that plants suppress weeds and not the crops, because you don't want to suppress both. There is not a general answer. As far as I know, you should look at the nature of the weeds. Some weed species form very little mycorrhiza, and their mycorrhiza has only very small benefits. So if then the crop forms more mycorrhiza and benefits more, then the mycorrhiza changes the competition between the weed and the crop, and that may result in the crop growing faster, which then outshades or outcompetes the weeds by which it's being culled. But there are certainly weeds that are as dependent on mycorrhiza as the crops, and there it is probably not possible to use mycorrhiza for weed suppression. So it depends very much on the nature of the weeds, but some of the more common weeds are not mycorrhizal, and there, just by changing the competitive balance, is probably a main mechanism. Because the fungus doesn't know what a weed or a crop is. And we classify plants as crops or weeds, but the fungus classifies plants as more or interesting partners to collaborate with.
Okay. Thank you, Professor Kuiper. So we really don't want to decrease the crop production, right? By applying mycorrhizal, we want to increase it.
But as long as your crop benefits more, and that's maybe a property of the traits of the crop compared to that of the weed, you can change the competition. And if the crop can better compete with wheat, you already have a higher yield and you have some form of weed suppression.
Thank you very much. Another discussion, probably some students want to ask interactively to Prof. Kuiper. It is once in a miles occasion to see Professor Kuiper from Wageningen University, since we are here usually find your regular lecture. Anyone want to ask a question directly to Professor Kuiper? Dina Hadi Soliha. I have insight from the committee that you want to ask some question for Professor Kuiper. I please you to ask the question directly. No one? You need to unmute your microphone first. Can you unmute your microphone first? Yes, it is unmute. It is unmute already. You can directly ask your question. I cannot hear your voice. Okay, or probably you can type your question in the comment room. Sorry, another student who want to ask directly to Professor Kuiper? Oh, okay. The question from Dina Hadi Soliha. So you have typed your question before. If the land is degraded, is it appropriate to use a non-mycorrhizal plant So what is the mechanism for providing the nutrients? Then if we want to cultivate on land that has almost recovered, can we combine arbuscular and non-arbuscular mycorrhiza? Can the two mycorrhiza be combined? Professor Kuiper, please answer the question.
Well, we did some work on forests in Africa, where forests are being cut and where the land is to some extent degraded. But it seems that in many cases there is still some mycorrhiza left. And so what is best probably in such cases is to use plants that are mycorrhizal, but that can still form, that can still grow reasonably well without it. Because if you use non-mycorrhizal plants, the soil will remain free of mycorrhiza. If you use a plant that can grow reasonably well without mycorrhiza, but still can form mycorrhiza, you can build up mycorrhiza for the plants to come up later. So I would say, yes, it is very important to select plants or to study the plants that come over there. And so plants that can grow without mycorrhiza or with little mycorrhiza are at the benefit. But if you have plants that are mycorrhizal, they can contribute to increasing the amount of mycorrhiza for subsequent plants. And that may be far more beneficial than having non-mycorrhizal plants, because that would keep the soil non-mycorrhizal.
One raised hand before. The committee, would you please do? Yes.
Okay, thank you very much for your time. Your presentation is really well, Professor. My name is Halbert. I'm from Research Center for Biomaterials, National Research and Innovation Agency of Indonesia. I would like to ask about the dual colonization. I heard that some plants have the ability to have two types of mycorrhiza, arbuscular mycorrhiza and also ectomycorrhiza in one species of the plant. In that case, I would like to, maybe you can give me insight how the mechanism, are they switching between them? One time they use arbuscular and then next time they use ectomycorrhiza, or they just use arbuscular and ectomycorrhiza directly on the same time? That's my question. Thank you very much.
Well, again, thanks very much for your good question. There are indeed a few plants that can form both arbuscular mycorrhiza and ectomycorrhiza. You can see them listed in the literature as what's called dual mycorrhizal plants. There are not many of them, but one major group is eucalyptus, because they can form both types. Mm-hmm. What people often thought was that they form arbuscular mycorrhiza in the seedling stage and only later form ectomycorrhiza. But that's probably not always the case. Mm-hmm. You can imagine that if a plant forms ectomycorrhiza and if the root is completely covered by this fungal sheet, you would not spend time in looking what's inside the root, even though there may still be arbuscular mycorrhiza. So they may be there. And then the question is, do they provide benefits? In some cases, they do. We know, as I said before, that the general effect of mycorrhiza is to increase the uptake of certain nutrients. But we also know that ectomycorrhiza is more efficient in the uptake of nitrogen. Arbuscular mycorrhiza is more efficient in the uptake of phosphorus. So there may be situations where plants can have benefit from both. But on average, and that's something you can probably imagine as well, if you have two types of mycorrhiza, you have to pay twice for the mycorrhizal fungus. So that's why it is something that is very rare, but it still occurs in some plants, and especially for the study of eucalyptus, this is very important. We have recently studied the ability of eucalyptus to withstand saline soils, or soils with too much salt. And there it seems that having arbuscular mycorrhiza is very important. We also did look a bit at ectomycorrhiza. We did not publish about it, but that is less important. So the ability to withstand salty soils is probably, and that may be one of the reasons why the eucalypts have maintained the ability to form arbuscular mycorrhiza. And of course, you can grow on more habitats, because if your seed lands in a place where other plants are arbuscular mycorrhizal, it may have a kickstart compared to plants that first need to see if there is enough ectomycorrhiza around. So you become a bit more flexible, but it is expensive.
I see. Thank you
It's
very much.
having two friends who do the same job for you at home.
Yeah. Thank you very much, Professor.
You're welcome.
Thank you very much, Halbert and Professor Kuiper. Probably two last questions. I see the question from Beauty Laras from Zoom chat room. Or probably you can directly ask to Professor Kuiper, Beauty Laras. You can just turn on your microphone and ask to Professor Kuiper directly. I please you.
Hello. Can you hear my voice?
Oh. No picture. Or the other one from Aditya Bagaswara. Do you want to ask your question directly, or I read your question?
Thank you, ma'am, for the opportunity, and good morning, Professor. My name is Aditya Bagaswara, and I would like to ask about how to differentiate between a mycorrhiza and a pathogenic fungi in several plants, especially on a rubber plant that once has a white fungi on its root that was called Rigidoporus microporus. That's my question. Thank you, sir.
What was the name you said in the end? Rizo?
Rigidoporus microporus.
Oh, Rigidoporus microporus. Well, that is a parasitic fungus, and so that is very different from the mycorrhizal fungi. That is to the best of my knowledge, but I think there are far better specialists than I am in parasites. I am not aware that there are specific studies how mycorrhiza could protect or reduce the damage by Rigidoporus microporus. So that is something that may need testing by checking different varieties of rubber, testing different mycorrhizal fungi, testing different conditions, how it would work best. But I'm not aware of any studies specific on that problem. But it could be a case where improved management could reduce the damage. But yes, parasites are also smart organisms. So if a plant is weak for whatever reason, they will always find a chance to enter. And rubber plantations, we know the plant is not indigenous in Asia. It came from the Amazon region, where it was brought by some doubtful means to Asia. And then you get very large monocultures, and we know that monocultures select for parasites much more than mixed cultures. And so we may also have to think about the specific growing conditions for such a plant. And so we also know that plants who move to a different area usually are much more sensitive to diseases because they have not evolved with them. So that may be another reason why that fungus is so much of a problem in rubber plantations in Asia, but not to the same extent to rubber plants in South America.
Does that answer your question, Aditya?
Yes, ma'am. Thank you for your explanation, Professor.
You're welcome.
And so the committee, it will be the last question, or Is there another? Okay. The last question from Fatahila Salsanuriski. Would you please to ask your question directly to Professor Kuiper?
Yeah, thank you for the opportunity, ma'am. For the professor, I am going to ask about fertilization time and mycorrhizal application time. Whether the application of mycorrhizae is carried out before, after, or at the same time as the application of fertilizer. Thank you.
Yeah. Well, I intend to speak tomorrow about the application and management of mycorrhizae in agriculture. There are discussions what is the best moment to apply mycorrhizae. Sometimes people apply them in the nursery. Sometimes people apply them during outplanting in the field. Sometimes they fertilize before or at the same time. You can imagine that if you add mycorrhizae and at the same time fertilize with high amounts of phosphorus, you would at the same time have a negative effect on mycorrhizae. So it depends on the nature of the fertilization when you could do it. But if you use mycorrhizae because you want to reduce your fertilizer level, then you should first think about your adapted fertilization in your mycorrhizal management. So just adding mycorrhizae plus fertilizer is probably not very efficient because of the negative effects of fertilizer on mycorrhizae. And I will show examples tomorrow how phosphorus fertilizer can have a negative effect on mycorrhizae.
Thank you very much, Professor Kuiper. Unfortunately, we are in the end of today's program, but don't be so worried because we will see each other again tomorrow at the same time and the same Zoom meeting link to learn about managing mycorrhizae in agriculture. Probably, Fatahila, you can follow tomorrow's guest lecturing to get a clearer answer for your question. I thank you very much to Professor Kuiper for your time and your great presentation, and also for all of the lecturer of Soil Science Department and all the students who follow this lecturing from both Zoom meeting and YouTube. I'm sure that we are here get so many knowledge and experience about mycorrhizal symbiosis. Before I close the program, I would like to request Professor Kuiper's time for a while for joining with us with the lecturer staff of Soil Science Department to discuss about core syllabus and potential collaboration in research and publication. And for all the students, please leave the Zoom meeting. Finally,
okay, and I'll see you all tomorrow. Thank you very much for your attention.
Thank you very much. Finally, I am Novalia Kusumarini, the moderator of today's program. As for leaving the meeting, wassalamualaikum warahmatullahi wabarakatuh, and good evening, ladies and gentlemen. And for the committee, please help to remove the students from our Zoom meeting.